Hello, Dr. Williams here with another revision video for you on the AQA biology content. This time it's from GCSE biology only, so that's the triple course. And we are going to look at B5 homeostasis and response. It's really important that you've looked at the combined video for this already. This is just building on that content, the extra knowledge that you need to know. So if you haven't already watched that, please go and watch that first. Otherwise, let's get going. So hopefully you'll remember that homeostasis and response is basically how we respond to those external things that are going on in our environment. So this is the definition of homeostasis, the regulation of the internal conditions of a cell or organism to maintain the optimum conditions. And that means that organisms and cells can respond to whatever is going on internally or externally. Now, if we have homeostasis when we think about body temperature, that means that our optimum conditions inside the human body is maintained at around 37 degrees. Remember from the work that you've done on B2, if the temperature gets too high, then the enzymes and the other proteins can denature. However, if temperature gets too low, then we have an issue with any enzyme-controlled reactions. For example, things like respiration will happen too slowly. So it's really important that our enzymes are able to function at their optimum temperatures. So we need to regulate body temperature. In order to regulate it, we need to detect the body temperature. So we monitor it and control it by a region in the hypothalamus in the brain, which is called the thermoregulatory center. And you can see the hypothalamus is tucked away right in the middle of the brain, just around here. And this thermoregulatory center in the hypothalamus contains temperature receptors. So as the blood passes through this thermoregulatory center, then these receptors can detect the temperature of the blood and decide how the body needs to respond in order to bring that back to the norm. Your skin also contains temperature receptors, and they're obviously going to detect the external temperature, but it's both the skin receptors and those in the thermoregulatory center that control your overall body temperature. And that's done through the sensory neurons and that communication that you learnt about as part of your combined science course. When our body temperature drops too low, we often call that hypothermia. And there's a number of responses that are brought about by that normal sort of response pathway that you're familiar with from the combined science content. The types of responses you get are these if body temperature is too low. You would get vasoconstriction. That is those muscles in the blood vessels near the skin constrict, so they become thinner. This allows the less blood to flow nearer the surface, so you will reduce heat loss to the environment. So that often makes your skin looking pale in colour because obviously you're getting less of that blood flowing nearer the surface. You also get the sweat glands closing, so there's no heat loss through sweating. You can actually, if it's quite extreme, start to shiver, and that's when your skeletal muscles start to contract and relax rapidly. And the reason they do this is because respiration is an exothermic reaction. So more respiration to contract your muscles means more energy is going to be released to the cells, which should warm them up because it's an exothermic reaction. And then obviously there are behavioural responses that organisms can do to respond to cold. We all put on a jumper, or some organisms will move to a warmer place or curl up or kind of huddle together to keep warm. If body temperature becomes too high, that is hyperthermia, and then you get different responses happening. Again, though, this is all muscle and gland controlled. So instead of vasoconstriction, we now get vasodilation. So those blood vessels, the muscles in them, the ones that are nearer the skin, will actually cause those blood vessels to dilate. That becomes wider. So more blood can flow to the surface through the capillaries, and they're close to the skin surface, so more heat is radiated away. Now, that kind of response often makes your skin look more red, which explains why your skin can look red when you exercise. Sweating will start, so you'll open the sweat glands and produce sweat. The sweat evaporates from the skin, as in doing so, transferring heat from your body to the surroundings, and that helps to cool you down. And then behavioural responses. There you can see a hippo. Hippos tend to stay in the water in the daytime to avoid the heat, and they come out and graze at night time. Okay, so there's lots of behavioural responses for different organisms that help them to control high body temperature. But the overall pathway of regulation of temperature, you can see it as a negative feedback loop. And remember, negative feedback is how the body responds to return the internal conditions back to optimum levels. And if you have watched the previous video, you'll see this normal or ideal level for a system is never stationary. It's never exactly the same. So if you're trying to get 37 degrees, it might be that your body temperature fluctuates slightly higher and lower than that consistently. Okay, but overall, these are the responses that your body will bring about. So if your body temperature increases, it's detected, remember, by those thermoreceptors in the hypothalamus and the skin. And the thermoregulatory center in the brain will coordinate that information and collect all that information and then send a response through muscle and glands, especially those that are in the skin. So you'd get vasodilation, sweating, and obviously behavioral changes to help bring back your temperature to the norm. With decreased temperature, it's exactly the same way of detecting it. So thermoreceptors in the skin and those in the hypothalamus will detect the change, the decrease in temperature. The thermoregulatory center, again in the hypothalamus in the brain, will respond to that and coordinate the muscles and glands, especially those in the skin, to bring your temperature back up. So by mechanisms such as vasoconstriction, the sweat glands will close, shivering, and certain behaviors. So this all helps to regulate temperature for humans at 37 degrees. But mammals and birds all use this mechanism and they have different optimum body temperatures depending on the species. Right, one of the other areas we need to look at is exactly how the brain works. So the brain, you'll have heard about, is quite an important sense organ in the central nervous system. And there are different regions to the brain which help it to carry out different functions. So there's the region called the medulla that you can see is in the main stem here. So you need to be able to identify where these regions are and what they do. The medulla, being in that main stem, has got really fundamentally important jobs. It's all our unconscious involuntary activities, which are we call autonomic responses. Things like breathing, those things you don't need to think about but are fundamentally important to keep you alive. That's most likely to be regulated by the medulla region. And then you've got the pituitary gland, which you should have heard about as being the master endocrine gland. And again, that is in the brain, just there, as you can see circled on the diagram. Then you've got the cerebrum or the cerebral cortex, and that really is about your conscious thought. It's a lot to do with language and all the thinking that you do, and your personality and all of those sorts of things. The thing that makes you you is in the cerebral cortex. Tucked away again in the middle here is the hypothalamus. We've just talked about the hypothalamus containing that thermoregulatory center, so it's a really important part of the brain with those receptors for that. And it also actually helps to regulate water concentration in the blood. And then you've got the cerebellum, and you can see the cerebellum just at the back of the brain. You can imagine it's a bit like a bell at the back of the brain, so that can help you to remember that that's the cerebellum. That again is important for some of the basic functions that we have, like posture, balance, coordinating our movement, as well as some roles in language and those fear and pleasure responses. So try to learn where these key regions are and have an overview of what each of those regions does. One of the paths of looking at this part as part of the homeostasis topic is to look at how we study some of these organs. So when you learn about triple, it's a lot of application questions. So one of the things about studying the brain is it's actually quite problematic. That is because of the risks associated with brain damage. It's hard to look at the physical kind of makeup of the brain without damaging it for the person. So most of the case studies from the past involve people who've been in accidents or illnesses that's damaged the brain. So things like strokes can be actually, quite useful to study to see how we can help people if they have a stroke in the future. And it helps us to understand how the brain functions a little bit more too. So to examine the brain's functions, you have to really try to study a working brain. That means you can't just do a dissection maybe on a dead body or a cadaver. We've got to actually start to look at how the brain works. So, and we also don't want to put anything invasive into the brain. So And we don't want to get scalpels and start to slice bits off. Obviously, that is going to affect somebody's brain function. So there's sometimes occasionally during medical procedures that might be necessary, but most of the time you cannot just study and have a little look at a working brain. If you do need to study a working brain and you do need to do anything invasive, like surgery wise, then often animal studies are the first port of call for this. However, to study the brain now, we really embraced the use of technology. So you can actually study the brain further by, as we said before, looking at brain damage. And there was this example of Phineas Gage, who had this pole sort of shoot through part of his brain and then went on to carry on living a life. But people were saying he wasn't quite the same person after that. So it was studying really documents from when that happened and what happened to his personality. That was useful. We've also got electrical stimulation, where you can put electrodes on this sort of cap and you can get a reading of how the brain works, maybe when you see certain pictures or have certain stimuli. And then we've obviously got scanning techniques. Some of you may have had some of these, things like X-rays and MRI scans and CT scans and PET scans, and they can all help us to have a picture of how the brain works and help us to see actually what is going on inside the brain just using these scanning techniques. The advantages of those sorts of new techniques like scanning is that we can start to look at how the blood moves through the organs and blood vessels in the brain. You can see any blockages and things like that. It can show swelling and inflammation as well as other 3D images, and it doesn't expose patients to radiation, so it's actually quite safe. However, the main disadvantages are that they are very expensive, so there are not many available, and you might have to wait several months to have a scan. The MRI scanners can be affected by movement. So when you're going to have one done, you've got to be very, very still while you go into the scanner. And that people can find that really uncomfortable, especially because while the scan is going on, it's in an enclosed space and there's lots of loud noises because there's some magnets rotated around you, and that can make you feel very claustrophobic. So there's obviously great advantages to using scanning, but also don't forget there can be disadvantages for using this technology. Okay, another sense organ that we'll explore when we're doing triple is the eye. And so the eye is your sense organ that contains lots of receptors which are sensitive to both light and colour. And the main parts of the eye, you've got to be able to label them, but also be able to explain their role in being able to see images. So the first bit that you need to be aware of is the cornea. And the cornea is the front clear bit of your eye. And that's the first place where light passes through, and there is some refraction or bending of light as it enters through the cornea. Then as the light enters through the eye, it goes through the gap, the space called the pupil, and then it passes through the lens, and the lens also refracts the light even more and alters in size to refract the light, so it lands right at the back on the retina. And the retina is basically your part of the eye that detects light and colour because of the receptor cells that are in there. There are two types of receptor cells, rods and cones. The cones are for colour, so think cones colour, and rods are for light intensity, black and white, really. Okay, they detect the light and they will transfer the information through the optic nerve as a electrical impulse, and it's the brain really that sees the image. So it interprets the image from those receptor cells that have been stimulated on the retina. The other parts of the eye you should really know are the iris, which is a coloured muscle at the front of the eye, and that can change to allow more or less light to pass through. You've got these ciliary muscles, which attach to suspensory ligaments, which then attach to the lens, and they contract and relax those muscles and pull on the ligaments to basically change the shape of the lens. And then you've got the sclera, which is the white part of the eye, which sort of protects the eye and everything within it. Okay, so how do we actually see and focus on objects? So focusing on objects is called accommodation, and this is a reflex mechanism that happens automatically to adjust the lens and focus the light onto the retina, and it can focus objects that are near or far. So as I said before, the cornea refracts the light a bit as it enters the eye, but the lens is the thing that changes its shape to fine focus the light, so the light lands exactly on the retina. And the shape of the lens can change because of the ciliary muscles at either side of them. Well, it goes all the way around the lens, really. We have to think of this as a 3D model. And they can contract. And when they contract, the suspensory ligaments actually loosen. When they relax, the suspensory ligaments are pulled tight. So we can see when the suspensory ligaments are loose, the lens bulges, so you get more refraction. And when they're pulled tight, the lens is pulled thin, so we get less refraction. So when we try to focus on distant objects, we want the ciliary muscles to relax. That means they move outwards and the suspensory ligaments pull tight and making your lens a long and thin shape. This means light is refracted less, so it can focus on distant objects onto the back of the retina. Okay. The opposite happens when we want to see near objects. Those ciliary muscles contract and they move inwards. This makes the suspensory ligaments loose, and the lens returns to a thicker spherical shape. So the light is refracted more because of that thicker lens to focus on closer objects. So this diagram is quite a nice summary to make sure that you try and learn. So to summarize the accommodation, if you think about distant objects, they need less refraction of light. So to get less refraction, you need the ciliary muscles to relax, suspensory ligaments pulled tight, and we need a long, thin lens. And there that diagram shows that. If you want to see near objects, the ciliary muscles contract. That makes the suspensory ligaments loose, and you get thick and spherical lens there, which refracts the light more. So if you learn that summary, then that should help you to explain how we see distant and near objects. Okay, we can also adjust the amount of light entering the eye, not only focus it, but adjust the light. So we need to adjust this so that the light doesn't damage the retinal cells. If the retinal cells do get damaged, it can lead to permanent eye damage. So it's very important that we get our iris muscles controlling how much light enters. And the iris muscles are muscles that surround the pupil. So you can see they are made up of circular muscles around the pupil, as well as the radial muscles around the first layer of the circular muscles. And these help to open the pupil so there's more light can get in and make it smaller. These two muscles are both under control in the autonomic nervous system. So this is an automatic response, and your pupils should dilate and get smaller depending on light or dark. And you can practice this just by closing your eyes. And then if you open them in front of the mirror, you can see that the pupil automatically adjusts to accommodate the extra light that they are now getting. So when the iris adjusts to the bright light, what happens is the inner circular muscles, the ones that go round in a circle around the pupil, they will contract, and these outer radial muscles relax. So working together, you can see that this reduces the size of the pupil. So then less light enters the eye. So as long as you remember circular contract, radial relax, smaller diameter, less light, hopefully you'll remember that. That's what happens when there's really bright light around. When there's dim light, when there isn't enough light, you're trying to see more. So the opposite happens. The radial muscles contract, the circular muscles relax, and the pupil diameter gets larger. So that means that more light enters the eye. Okay, so that obviously you want a bigger pupil to get in more light, so you can detect that by your retina. So again, there's quite a nice little summary here. If you remember that the amount of light, when there's more light, the muscles contract and the inner circular muscles, that means they'll move inwards. Outer radial muscles relax and the diameter pupil is smaller. Less light, those inner circular muscles relax and move outwards. Outer radial muscles contract and the diameter of pupils get larger. So learning that summary, again, if you're asked to describe how the iris works, you should be able to put those into words. Now, obviously, we know we have problems focusing on the retina. If any of you ever wear glasses or contact lenses, then that is because you're not quite focusing on the retina accurately. So with normal eyesight, the light can be focused on the retina. You can adjust your lens and make sure that you've got a clear sight of whatever you're looking at. Short-sighted people, it's called myopia. And for whatever reason, because of differences in their eye, they can see near objects clearly, but distant objects are blurred. And that's because they are short of the retina when they're trying to focus. So you can see that the focal point is not quite reaching the retina when they are looking at distant objects. In long-sighted people, which is called hyperopia, they can see distant objects clearly, but near objects become blurred. And that is because their focus on near objects is going beyond the retina. And it can be due to the shape of the eyeball, but it also can be due to things like the lens not being able to adjust properly to get the light focusing on the retina accurately. So if you've got some problems with focusing on the retina, what can happen is that you can easily correct it using spectacle lenses or glasses. If you're short-sighted, which is myopia, remember, you can correct this with the concave lens. So lens is caved in or thinnest in the center. This makes the light rays more divergent, so they spread out before they enter the eye, and this reduces the eye's focusing power, moving the focus point backwards onto the retina, so it relieves that lens from having to focus the light as much. So concave lens are to correct short sight, and if you remember that, that's because it's short in the middle. If you think about long-sightedness or hyperopia, these can be corrected with a convex lens. The lens is thickest in the center. They converge the light rays, bending the light inwards before it enters the eye. So it's going to bend that light a bit more. This increases the eye's focusing power, and you can get the focal point moving onto the retina, so it doesn't go beyond it. So if you try to remember this by convex lens to correct long sight, and that is longer in the middle of the lens. There are other methods, obviously, of correcting vision. Contact lenses, again, they can use those ideas of convex and concave lenses, but they are tiny lenses that actually sit in contact with the eye. You can also have laser eye surgery, and laser eye surgery can use to flatten the cornea, and that helps to correct short-sightedness, or to even increase the curvature of the cornea, and that helps to correct long-sightedness. So to just reshape the cornea, so that helps with the refraction of light, just to make it focus on the retina correctly. And you can also have lens replacements. So some diseases like cataracts, the lens clouds over. It becomes filled up with minerals, so it makes it cloudy and you can't see properly. So you can actually have a replacement lens implanted into the eye, which actually is responsive to the muscles around it and can focus onto the retina correctly and automatically. Okay, the next phase then, we've looked at the brain and the eye and control of temperature. You also need to look at maintaining water balance. And it's really important that we maintain water balance. This directly links to osmosis. So make sure you understand the definition of osmosis. If a body cell loses or gains too much water and we get osmosis, they don't function efficiently. So for example, in our red blood cells, when water moves out of the cells from a dilute, which would be the cytoplasm, to concentrated solution through a post-pulmonary membrane, that would be an example where you've got high glucose concentration in your blood and things like that, and water might move out of the cells, okay? If there's no net movement, that's the ideal scenario. We don't want water moving in or out, so everyone's happy, the cells are happy, we are regulated. But when water, if your blood is too dilute, then water will move out of the cells, into the cells, and those cells will end up bursting. And so then you have to be able to describe for all three scenarios what is going on in terms of osmosis. So although osmosis is paper one, make sure you do understand it for paper two. Okay, so how do we regulate our water balance? So water, ions and urea constantly being lost through the body. Whenever we breathe out, we're losing water. Our skin, in our sweat, we excrete water, ions and urea, and in our kidneys, we excrete water, ions and urea. So we have to regulate this carefully. What we do is the blood is regulated by the kidneys and excreted as urine. There is no control over that water loss in iron or iron loss in lungs or your skin. It's all down to the kidneys to regulate the losses of these ions and water. And we need to balance the amount of water in our body at all times in order to regulate and maintain homeostasis. So we, on average, take in about 2.5 litres of water a day. We will make 200 mils from metabolism, 700 mils from food, and 1,600 mils from drinks. and we excrete the same volume of water out. So our lungs around 400 mils, sweat around 600 mils, and our kidneys around 1,500 millilitres of urine a day. And in that urine, we get excess water, ions, and urea removed from the body. If you remember that urea is actually made from waste amino acids, they are taken to the liver and transported, broken down in there into urea and transported through the blood to the kidneys, where it's excreted through the urine. So it's just remembering this again from paper one, that amino acids, when they are transported through the blood to the liver, they are going to be broken down, any excess ones, and converted first to ammonia, but then form urea. And that urea then is in the liver and it goes through the blood to the kidneys and it's excreted out of the body. So this is a toxic chemical that we don't want in high amounts in our body. So overall, this is what the kidneys are doing. The blood is entering the kidney and it's being filtered. When the blood leaves the kidney, it's got less ions and less urea and continues around the body to carry out its function. What's been filtered out of the blood is called the filtrate, and that will become the urine, and that has got the water, ions, and urea in. This urine will pass through a tube called the ureter into the bladder, and the urine in there is stored in the bladder until it's excreted out through the urethra. So the kidney filters the blood. If you can imagine that blood is entering the kidney with all of its red blood cells and white blood cells and platelets, the proteins, glucose and urea, water and ions, by the time the blood comes back out of the kidney, it's still got those cells, it's still got proteins, it's still got glucose, but it's just got less urea, less water and less iron, so that it's regulated those things. The urine that's produced can have any excess water that the body might have, any excess ions that it might have, and it also has urea in there as well, as much as urea as it can excrete. If we look at the structure of the kidney, inside the kidney themselves are these little tubes called nephrons. And these nephrons are responsible for filtering the blood and making that filtrate, which becomes the urine. The filtrate, so that's any substances that have been filtered out of the blood, goes in the nephron, stays in the nephron, and is adjusted according to how much water we need to reabsorb and how many ions we need to reabsorb. And then the urine goes down through the collecting duct and then into the ureter to the bladder. Okay. So these nephrons have a function of like 3 different stages and how they work. Stage 1 is the filtration stage, where the blood is passing through this capillary and it goes into this part of the nephron. You don't need to know the specific names of the parts of the nephron, but you just need to know that blood is going through that capillary in high pressure, and that high pressure causes urea, water, ions, and even glucose to be filtered out. The blood cells stay in the blood. Any large molecules, such as blood proteins, are going to be too big to fit through the capillary lining, so they stay in the blood and they keep travelling around. The second stage then is to reabsorb things that you still need. So the kidneys have first of all filtered lots of things out, but they've also filtered things out like glucose, which is a small molecule, but you still need it. So the kidneys selectively reabsorb any molecules the body needs back. So they take all of the glucose back. They're going to take as much water as the body needs and as many ions as the body needs. And then the final stage is just adjustment of how much water, urea and ions that we're going to put into our urine. And so that's called the formation of urine. And this is where we get a fine tuning of how much water we're going to reabsorb. The urine then passes into the bladder, and then the kidney has now regulated the water and iron content of the blood, and it's kept urea levels at a low level. So the mechanism by which the amount of water and iron is controlled is by a hormone called ADH. And ADH is actually produced by the hypothalamus. And the hypothalamus is all the way in the brain, but the hypothalamus produces the hormone ADH, which actually regulates the water levels. So although the hypothalamus produces it, it's the pituitary gland that releases it. So the hypothalamus actually has receptors that detect the water level and makes the ADH. It's constantly making ADH actually, but it decides how much pituitary gland is going to secrete ADH. So the hypothalamus is not a gland, which is why it doesn't secrete ADH, because only glands secrete hormones. ADH is always present in the blood, it's always released, it's just the concentration of it alters depending on the water levels. So ADH effectively works by affecting the permeability of the collecting duct. So remember, the collecting duct there has the water and ions and the urea that are going to make the urine. But when you've got ADH present, it's going to affect the permeability of the collecting duct to allow either more water to be reabsorbed into the capillary or less water to be reabsorbed back into the body. So it can regulate and work out how much volume of urine is actually produced. So the way it does that is actually quite complicated, but you need to really know that that the way this whole thing is coordinated actually can really regulate the urine amount produced and the volume produced of that urine. So you can see as a summary, that's rubbish. So here's a summary of how that works. So when your water is too high, you've got a dilute blood. And so receptors in the hypothalamus detect it. The pituitary gland secretes less ADH into the blood and less water gets selectively reabsorbed into the blood from the kidney. If you look at water concentration in the blood is too low, so your blood is quite concentrated, receptors in the hypothalamus again detect it. The pituitary gland actually secretes more ADH and more water is selectively reabsorbed. So if you can remember that less ADH means less water reabsorbed, then you will actually produce a large volume of light coloured urine, because remember, less water is being reabsorbed back into the body. If you can remember, when water is too low, the pituitary gland secretes more ADH, so more water is reabsorbed, then you'll get a smaller volume of dark coloured urine. That's my top tip, really, to remember: less ADH, less reabsorbed, more ADH, more reabsorbed. And then hopefully you can work out the other bits around it based on those two kind of key ways of remembering it. So here's a diagram that really shows you what is happening in the collecting duct and the capillary. So you can see in normal levels of water, normal levels of ADH are released, and the ADH will bind to these receptors here in the collecting duct on those cells, and you will get the normal permeability of the cell. So normal water levels are reabsorbed and a normal volume of concentration of urine is produced. However, when you've got high levels of water, so you've got a dilute blood, you want low levels of ADH. So again, they will bind to those receptors, but at a lower level, which means that these permeability of these membranes is much less permeable. So you get less water reabsorbed and you get a larger volume of low concentration urine. And in contrast, if you've got low levels of water, you get high levels of ADH being released by the pituitary. They make membranes much more permeable when they bind to all of those receptors. So you get high levels of water reabsorbed from the collecting duct, and you end up producing a smaller volume and a high concentration of urine. Okay, so it's just a question of remembering the mores and the less, and trying to kind of visualize what's happening in the collecting duct and the capillaries. Now, if the kidneys go wrong, you can treat it. People who suffer from kidney failure might be treated with an organ transplant or using dialysis machine. A dialysis is an artificial kidney. The dialysis fluid will have similar concentration of solutes to the blood, and your blood actually is basically shunted around through the dialyzer, and then the cleaned blood is returned to the body. And when it's in the dialyzing machine, it will remove any excess ions and water. And you have to go a few times a week to have this done in a hospital, and you can stay on a dialyzing machine for many years, but eventually you will need a kidney transplant. There's obviously risks of infection and things like that with having dialysis, so it isn't a long-term solution. If you need a kidney transplant, you can actually get that from a living donor, because you only need to survive one kidney will allow you to survive a perfectly normal life. So you can actually get a kidney transplant from a living donor. Okay, the last part of this is to look at plant hormones, and plant produce hormones to coordinate and control growth in response to stimuli, just like animals do. And plants use light to carry out photosynthesis and use the glucose to make proteins for growth. So if you're ever asked how do plants grow, your answer needs to be structured around, because they use light for photosynthesis, so they can produce the glucose and use the glucose to make proteins for growth. Make sure you're linking those keywords if ever you're asked how plants grow. Plant hormones are really important throughout all the stages of a life of a plant. So at seed level, you can see that's the seed and acorn. When the seed starts to grow into the plant, we call that germination, and a seedling will start to grow. And then obviously growth at later stages into the full oak tree. When we talk about plants growing towards a stimulus, we could call that a tropism. And if it is towards that stimulus, it's a positive tropism. If it's away from a stimulus, it's a negative tropism. So plants can actually grow from seeds in response to light, which we call phototropism, gravity, which is called gravitropism, and water, which is called hydrotropism. So you can see those come from their Latin names: photo, light; gravitropism, gravity; and hydro, from water. And then if we look at each of those tropisms in turn, the roots and the shoots can respond differently to them. So obviously, if you're a plant and you're starting to grow, your roots are going to want to grow towards water. So the roots of a plant are positively hydrotropic. That means roots will grow sideways or upwards or wherever the water is, so that it can reach water. They have a stronger response to water than to gravity, so the plant always ensures it gets whatever water it needs as a priority. If we look at gravitropism, we'll see that the roots are positively gravitropic, growing towards gravity, but shoots are going to be negatively gravitropic, and they grow away from gravity. This is why a plant, no matter which way up you put the seed in the ground, always grows with shoots up and roots growing down. And then the last one is phototropism. So shoots always grow towards the sunlight. They are positively phototropic, and roots grow away from sunlight. They are negatively phototropic. Again, this helps to explain the direction of shoots growing up and roots growing down, no matter what way you put the seed in the ground. So Darwin was actually a scientist who investigated this phototropism, so how plants respond to light. And he did a very simple experiment where he covered the tip of a shoot, and he covered the middle of a shoot and the bottom of the shoot. And what he found was the ones with the tip that were covered grew straight in response to light coming in from the side. However, the ones that didn't have the tip covered grew towards the light. So from this, he concluded that actually it was the tip of the plant which was sensing where the light was coming from. Further experiments showed that this growth is caused by a hormone called auxin, and that is actually produced in the tips of the shoots. And the tips of the shoots make this growth hormone, and the auxin will cause cells to elongate and grow by cell division. So if you can see this diagram, it shows that auxin made in the tip when the shoot was covered will diffuse evenly down the shoot, which is why you had straight growth. But those with the tip uncovered, the auxin was produced in the tip, but it diffuses unevenly down the shoot. So we get phototropism shown with the shoot bending towards the light. If we look at that in a bit more detail and do further investigations, what we find happening is that auxin is made in the tips of shoots, and it diffuses unevenly because more auxin always goes to the shaded side of the tip. At this side of the shoot tip, then you'll get more cell elongation and growth, which means that the shoot will start to bend towards the light. So this is how auxin works in the shoots. It goes to the shaded side and causes elongation on one side more than the other. Gravitropism, though, is slightly different. When you do investigations on gravitropism, you put germinating seeds in different positions and leave them to germinate. What you find from that is it's still the growth hormone auxin, but it actually has a different response in roots than it does in shoots. There is again an uneven distribution, but this time the auxin moves in response to gravity, and it goes on the lower side of the roots and the lower side of the shoots when you first put them in. But in the shoots, it causes more cell elongation, so the shoots grow upwards. However, in roots, the auxin on the lower side causes less cell elongation, and that causes the roots to grow down. So it is a bit confusing, but as long as you remember the responses in shoots basically causes more cell elongation, and it goes on the shaded side or the lower side in the shoot when it's gravitropism, then you're going to get hopefully a good understanding of how they respond to auxin. And if you remember in roots, there's a different response and it causes less cell elongation. Hopefully you can work out any challenging questions around this. There is a required practical to go with this investigation. It often includes growing seeds and getting them to germinate, and then thinking about things like your sort of variables that you might have for this investigation. For example, how many seeds to grow, how much water to give them, and then thinking about how you can measure the length of each seedling and where your light stimulus might come from. So this phototropism investigation, you look at germination really and give them light from different directions. Just don't forget that actually measuring seedlings is quite a tricky thing to do, especially with a ruler. So you can use a bit of string to measure the length, and that might be more accurate, and then straighten out the string onto a ruler. So that might be a more accurate way of measuring seedling length. And it's important to do a lot of repeats for this investigation because of all of the variables involved with growing seedlings. Other uses of auxins, we can start to think about auxins responses in the plant and how to basically use them as scientists and gardeners to help us grow our plants more effectively. So some other uses are as rooting powders. We can actually take a cutting from a plant and put it into rooting powder and then get the auxins, because it stimulates growth, to make more roots grow. So it helps us to grow plants much better. Rooting powders are also used in cloning. So cloning, you can scrape some seeds from a plant and actually separate out those sort of the scrapings, and they will grow into individual little seedlings. So it's a rapid way of cloning, and you grow them on agar plates, and in that agar plate you've got rooting powder, which helps those to start to produce roots and quickly grow. So that can help to promote the growth in tissue culture. And you can also use auxins as weed killers because they're a growth hormone that make things grow faster. They actually are used in weed killers because they get plants to grow so quickly that they can't actually keep up with the energy demands that they need. So they can't release enough energy, they can't photosynthesize quick enough, and the weeds actually end up dying. There are other plant hormones too that can be used. Ethene is an example of one. This is sprayed onto fruits to help them store and transport fruit. So bananas can be harvested before they are ripe and then reduce the damage when you're transporting them and store them. And then when they're ready to be sold in the shops, they are sprayed with ethene to ripen them just in time. And there's hormones called gibberellins, and there are three main uses of gibberellins, and you can use them in agriculture and the horticulture industry, and they are to end seed dormancy. This is when germination happens, to promote flowering, so flowers bloom when you want them to, and to increase the size of the actual fruit that you want. So, for example, grapes. Okay, so that should be all of the content for this. Hopefully it's not been too quick for you. Feel free to go back and have a watch over a few things again if you need it re-explaining, but good luck with your exams coming up.